Hi, Year 11s. Welcome to week 3 of our PowerPoints in cells. Today we're going to be looking at cell division, which means we're going to be looking at binary fission as well as mitosis. In stage 1 we don't look at meiosis, that's the third type of cell division that we will look at in stage 2. So today just going to be looking at binary fission and mitosis. Before we start talking about that, we do have to talk about the duplication of DNA. So basically, all cells before they divide, they need to make a copy of their DNA through DNA replication. Going back a step, we know already from our research and our learnings the last 2 weeks that all cells contain some sort of DNA, and that DNA is made up of a nucleotide unit, and that nucleotide unit is made up of a phosphate group, a sugar, and a base. The sequence of the bases in that DNA will form what's known as codons, and they're triplet codes of the bases there. And those codons will allow the cells themselves to make proteins that they need for metabolism to survive, etc. Two different types of cells, we have prokaryotes and eukaryotics. In each case, DNA is stored differently in those cells. So in eukaryotic organisms, the DNA is stored in the nucleus. You have more than one chromosome, and they're also in a linear fashion, whereas in prokaryotic cells, there's only one chromosome, there's no nucleus, the DNA is stored in the region called the nucleoid, and it's also circular. Regardless though, both prokaryotic and eukaryotic cells, before the cell divides, it has to go through DNA replication. What happens there is that the molecule of DNA is duplicated and the chromosome makes a copy of itself. You then end up having two identical copies that are called chromatids, and they're joined at the center through a centromere. So this shape here, the chromosome here, this is when DNA replication takes place, and this is only time, and this is a eukaryotic chromosome because it's linear. This is the only time that a chromosome actually looks like this because it's gone through DNA replication. So the yellow might be the old strand, and the blue is the new strand that's been created, and they're held together by a centromere. So this still counts as one chromosome. Even though there's double the DNA, it still counts as one chromosome, and it's not until the cell physically divides and one of these chromatids will go to each side or each cell that the chromosome number actually doubles. And even then it's not going to double because then each new cell will each have 46 chromosomes if it's human, for example. So even though the DNA is replicated, it's still only one chromosome, but you have doubled, so instead of having one chromatid, you now have two chromatids. Talking a little bit more about cell division though, so cell division is really important for basically 2 of the 7 processes from if something's living. So Mr. Sheng, if we go back to that in the future, and we talked about that again in class this week. So basically what cell division is, you have a parent cell, and that parent cell divides into 2 new daughter cells, and those daughter cells will be genetically identical to the parent cell, and that's because that the DNA has been replicated and each daughter cell gets an exact copy of that duplicated DNA. That's important because without that we can't have growth and reproduction. So in eukaryotic cells it's mostly going to be for growth. For mitosis especially, mitosis is going to be specifically for growth, for the organism to grow itself. It might be to repair or replace damaged cells, but most of the time it's going to be for growth. But it can be used, cell division, in the form of meiosis for reproduction in eukaryotic organisms. In prokaryotic cells though, cell division is generally for reproduction because they're only a one-celled organism. Every time they divide, you're duplicating the amount of organisms that are there. So if a bacteria divides, instead of having one bacteria, now I've got 2, so it's for reproduction. But I suppose you could also look at that in terms of growth, that the population number has grown, so the population has experienced growth itself there. So you could kind of look at it as both ways there, but generally cell division in prokaryotic cells is for reproduction. Most of the time in eukaryotic cells it is for growth, and that's when we're talking about mitosis. But if you go through meiosis, that's going to be reproduction for eukaryotic cells too. Let's talk about this because cell division does look different in prokaryotic and eukaryotic cells. We're going to start focusing on prokaryotic cells, and remember we're only talking about mitosis when we go to eukaryotic cells, but first binary fission for prokaryotic cells, and we've got binary fission happening here. So basically binary fission actually just means dividing in half. And most bacteria, in fact all bacteria that we know of, they consist of only one single circular chromosome. That singular chromosome has several hundreds of genes, and basically because of this, prokaryotic cells only got one chromosome with several hundred genes, whereas eukaryotic organisms have a number of chromosomes with thousands upon thousands of genes. cell division is a lot simpler in prokaryotic cells, and it's only a 4-stage process. So 4 stages, as you can see here. Very simple process. DNA has to be replicated first, which we can see has happened here, and then the division itself will take place. So what are the 4 stages of binary fission? So first off, the chromosome is replicated, produces 2 identical copies of the DNA. So we can see here we have DNA in the nucleoid. Now the DNA is replicated, and we've got 2 copies of that. Both copies of that chromosome are attached to the parent cell's membrane, so that you can't see in this diagram, but it will actually, the chromosomes will both attach to the sides of the membranes, and the cell will start to grow in size. And as it grows in size, the chromosomes are actually physically separated because they're each connected to the membrane's being stretched out. One chromosome goes towards this side with the new cell, and the other side goes this side to the new cell. Eventually, this puts so much strain onto the cell membrane that the cell membrane physically rips in half, and you end up with 2 daughter cells, and the chromosome will unattach from the cell membrane back to the nucleoid region. It's a very simple process. DNA replicates, DNA attaches to the cell membrane, cell grows in size, cell divides, one chromosome in each cell. Very simple stage for binary fission. Mitosis, on the other hand, mitosis is a bit more complicated. Again, as I said before, this is more complicated because they have organelles in cells that need to be replicated. It's not just the chromosome and the cell itself. You need to replicate all the chromosomes itself. There's more DNA and there's more chromosomes. And the chromosomes in humans in humans is made of something called chromatin, which is a mixture of DNA and protein. So it's not just DNA we're dealing with here. So it's a lot more complicated process. So first off, before we even talk about these phases, let's talk about what the phases are. So there's basically mitosis can be broken into 2 stages, mitosis and cytokinesis. So mitosis is the first step. It's the breakdown of the nucleus. So when we're talking about cell division, mitosis is actually, so mitosis is made up of mitosis and cytokinesis. That mitosis stage itself is the division of the nucleus and creating a new nucleus, whereas cytokinesis is the physical separation of the 2 cells. So mitosis, breaking down the nucleus, separates the sister chromatids into single-stranded chromatids, and cytokinesis, the physical separation of the parent cell into 2 daughter cells. So mitosis itself consists of these 4 steps here, so prophase, metaphase, anaphase, telophase, and then you've got cytokinesis in its own stage down below. So, easy to remember, PMAT-C. PMAT-C is how you're going to remember the stages of mitosis, and you do have to know those names, so it is worth remembering them. We're going to look at each stage. You haven't had to draw these so far, but I would recommend moving on, starting with this slide, that you actually draw the stages of mitosis. So starting with prophase. Now, I have little tricks to remember some of these stages. So for prophase, it's the first stage of mitosis. I remember it's the first stage of mitosis because it starts with the prefix pro. If someone's a pro or professional, they're really good at what they do and they might come first in a competition. So prophase is the first phase of mitosis. During this stage, our chromosomes will condense, so that means that the DNA starts to coil up really tightly, it takes up less space inside the chromosome, but at that point they become more visible under a microscope. Then we have the nuclear membrane here. The nuclear membrane starts to break down and the chromosomes are released into what's known as the cytoplasm of the cell. There's also some changes happening in the cytoplasm of the cell. What's also happening in here is we have centrioles, which are cylindrical structures of protein. They're going to start to move, so that's here, they're going to start to move to opposite sides of the cell, and they're going to produce something called microtubules, specifically spindle fibers, which are made of microtubules, and they're going to attach to the chromosomes a little bit later on. So that's what's happening in prophase. I would be able to draw this. Then we move on to metaphase. So metaphase is the second stage of mitosis. Now in metaphase, first off, the main thing here is that the chromosomes are lining up down the middle of the cell. Now, for me, I remember that because metaphase starts with an M and so does middle. So chromosomes lying down the middle, that's what happens in metaphase. What's actually happening in the whole process though? So the centromeres of each chromosome, they're being attached to the centrosomes. Okay, so spindle fibers coming out and it's attaching to centromeres, and then the spindle fibers are going to start to contract and they'll move the sister chromatids to sister chromosomes to the center of the cell. So if you think about it in terms of how it works. Say you had 2 people at the end of our classroom, and then there's a third person just walking around the room back and forth. Both people at the end of the classroom have a lasso. They throw them around the person that's walking in the classroom, and they both pull with equal strength. Now what's going to happen is, because of the tension coming from the string and the length of the string being the same, that person's going to end up in the dead center of those 2 people pulling, and that's what's happening here. We've got our spindle fibers attached to the chromosomes. They're both pulling with equal force. Sorry, I've changed the picture of the side there. They're both pulling with equal force, and therefore we end up with all the chromosomes lining down the center. Now we call that the equator of the cell, because the cell, in this analogy, the cell is like the Earth, but it can also be called the metaphase plate. Next up we have anaphase. Anaphase is when you're going to have spindle fibers pulling the sister chromatids toward the centrioles at each pole, so they're continuing to pull, but this time there's nowhere they can go. The chromosomes are already in the dead center of the cell, there's nowhere else they can go, so as the spindle fibers continue to pull, what happens is the centromeres will actually be broken, and instead the sister chromatids will move. One sister chromatid from each chromosome moves to each pole. Basically then what's going to happen is the cell will start to elongate, which means it starts to get longer. I don't have any tricks to remembering anaphase, unfortunately, so prophase and metaphase remembered, and now it's just we remember anaphase. Lastly, for mitosis, we end up in telophase. In telophase, now that we've got our chromatids on each side of the cell, we have a nuclear membrane starting to form around each set of chromosomes. So at this stage, we're going to end up with 2 different nucleuses inside the cell. And essentially the chromosomes are no longer going to be condensed, so we can no longer see them under a microscope. Cells continuing to elongate. Now that we've finished mitosis, though, we do move into the stage called cytokinesis. Now in cytokinesis, we have to go back to anaphase for a second, because in anaphase it's also important to note that during anaphase 2 proteins have assembled at the equator of the cell while the chromatids are being separated, and that's actin and myosin. So in this stage here, along the equator of the cell, we have actin and myosin starting to appear. Now what's happening here, these 2 proteins, they form what's known as a contractile ring that goes around the membrane, and it starts to squeeze the center of the cell as it condenses, and it starts to smaller and smaller and smaller, and as it does that, it pinches the cell membrane into what's known as the cleavage furrow. And it keeps doing that. The contractile ring keeps condensing until the cell is physically ripped in half. The membrane here will close off with each other and form 2 individual cells. At the end of this process, each daughter cell will be identical. So the cells produced will be identical. They're basically clones of the parent cell. They'll have the same number of chromosomes and the same type of chromosomes as their parent cells, and it ensures that all of the cells that are created are going to be known as diploid cells. They have 2 sets of chromosomes. Lastly, what we need to do this week is we need to look at the differences and similarities between binary fission and mitosis. So, similarities. In both processes, chromosomes are separated only after DNA is replicated. Both processes are going to be used for growth and reproduction. Both processes include 1 parent cell splitting into 2 daughter cells, and both are forms of asexual reproduction. So if you're ever asked in a test or an exam what's the similarities between mitosis and binary fission, there's your similarities. But there's also some very key differences over here. So firstly, the binary fission is only happening in prokaryotic cells. Mitosis is happening in eukaryotic cells. Prokaryotic cells are going to reproduce in 1 stage, binary fission, whereas eukaryotic cells are going to go through mitosis and cytokinesis, so 2 stages. Chromosomes themselves will be separated by the cell membrane in prokaryotic cells, whereas in mitosis by spindle fibers. And lastly, mitosis is that more complex process because of the organelles, because there's more chromosomes. It's much more highly organized, it's broken down into more defined steps, and it's just a more complex process. That's it for this week. I've gone through that very fast, so like I said, we'll cover that in class. We're going to do some experiments where you're looking at all of the stages of mitosis and hopefully binary fission under a microscope. Please ask lots of questions in class to help cement this, and we'll be working on, I think it's 1.3 in your textbooks, cell division. See you in class.